My name's Emma Martin. I'm a biology major, marine science minor, and I've been working in the Randy Rogen Marine Bio Lab here at BU for the past 2 years. And for my senior thesis, I'm investigating whether larval fishes are exhibiting a distribution shift in response to El Nino Southern Oscillation climate events in the Central Pacific. So this is a view of the Pacific Ocean. Absolutely massive, covers about half the Earth. And there's so much that goes on here, so much primary production, so much biomass production, nutrient cycling, species diversity, that's incredibly important to understand the inner workings of this system. So the main driver of climate events in the Pacific is the El Nino Southern Oscillation Cycle. So during ENSO neutral years, what we see are really strong easterly trade winds blowing surface waters, the warmer surface waters, westward across the Pacific, where they build up into this warm pool. And that allows for more upwelling and nutrients to be brought up to the surface along the South American coastline that then spread across the Pacific. So during El Nino conditions, we see a weakening of those trade winds and the warm water pool spreading back across the Pacific. And there's a decrease in primary production and nutrient availability as well. And then during La Nina years, we see an opposite trend where there's further spreading of this cool water pool on the South American coastline, more primary productivity, more nutrient availability. So the ENSO cycle overall consists of an El Nino event, a La Nina event, and then ENSO neutral years, which typically repeat every 3 to 8 years. And we've seen an increase in frequency and intensity of El Nino events over the past several decades. So there are definitely some big changes shifting in the cycle, and it's going to be really important to understand how it's affecting populations in the ocean. So developing fish eggs and larvae are extremely buoyant because they have a very high lipid content. So they spend these really delicate developmental phases right on the surface of the ocean. So as they're so delicate, they're extremely susceptible to any changes in sea surface temperature, nutrient availability, and it definitely affects the viability of the populations overall. So my study area is the Phoenix Islands Protected Area in the Central Pacific. It's this box here. And it's under the governing body of the Republic of Kiribati, which is comprised of several archipelagos outlined in white here. And the Phoenix Islands is the largest and deepest UNESCO World Heritage Site. It's a large California-sized MPA, and it provides a really unique opportunity to understand the impacts of El Nino alone on these fish populations, as it was closed to extractive fishing efforts in 2015. So what this diagram shows on the left here is fishing effort prior to closing, and each light here represents a boat or a fishing effort. And you can see the difference, the actual impact that was made after this was closed. So this is huge. And this area has always been known as an extremely productive area for fisheries like tuna, anchovies, the very large-scale Pacific industries. So what I'm trying to do with this study is, first of all, identify which larval fishes are actually present within PIPA and get an idea of our biodiversity, get an idea of population numbers. And then additionally, I want to see how these ENSO states are affecting spatial distributions of these fishes and their groupings or assemblages together. So that's just basically whether some fish families are found together consistently and how that changes over the years. So we've partnered with Sea Education Association, which is a student-based sailing and learning platform. And each summer, they go out to the Phoenix Islands and actually take these plankton samples. And our lab has been sending someone to the Phoenix Islands as well over the past years to take part in this. So what they do is they drag these plankton toes, these net toes, through the surface waters and collect all of the biomass, so all of the small plants and animals that make up the biomass in there. And we get something like this. And so for a semester or two in the lab, I was one of several students whose job it was to sort through these jars of biomass we'd get, and we would pick out every individual fish larvae or fish egg. And we still have so many people helping out with that because my data is just the first 3 years of a 10-year study, so it's an ongoing process. And what we want to end up with is a plate of just the fish eggs and larvae. And then we're going to explore that a little further as well. So what the graph on the left here shows is the past 80 years of data for sea surface temperature anomalies associated with ENSO. So what that means is anything in red here above the x-axis is a deviation from the norm higher, and then blue is a deviation lower. So the bright red box represents extremely strong El Nino events, which is my peak year for 2015, and then below are La Nina events. So my peak here for 2016 is a weak La Nina event, and then 2017 is an ENSO neutral year. And you can see on the right, I've done an inset of that data to blow up more specifically, and these purple boxes represent the actual time frame that our data were taken in. So towards the beginning of a very strong El Nino from 2015, La Nina during a weak La Nina in 2016, and then 2017 was ENSO neutral. So there are a lot of environmental variables that are shifting as we go through here. We have sea surface temperature with a drastic shift from very hot to very cold, and then back towards a median for ENSO neutral. We have salinity with a dramatic shift from El Nino to La Nina, and then I've also tracked moon phase as well to see if that's a variable that's affecting these populations. And my fourth and final variable at this point is chlorophyll A, which is a metric for primary productivity. You can see there's hardly any in El Nino, where La Nina and ENSO neutral years have much more primary productivity. So this study is unique in that it has a year from each ENSO state, so it really is a full slate to look at. So for my methods, as I mentioned, we collect all the ichthyoplankton, which are fish larvae and their eggs, and then what I actually did is looked at each one under the microscope and taxonomically ID'd it to the family level using some books or ID guides that show, how many rays are in this type of fin and what's the head shape like. And I built a photo reference library for that so that the study will have that as it continues, and we're confirming some unknowns with DNA barcoding. So I identified 10,854 fishes from a minimum of 43 families, and less than 0.075% of those are unknowns. But this is it was a lot of data wrangling for this project, but the important part of this slide is the actual end values for the years. So we had 3,400 fish in the El Nino year, 4,300 in La Nina, and 3,000 in ENSO neutral. So a big difference. So I wanted to say, how does overall fish family abundance change with ENSO state? On this graph, my taxa are aligned on the left, and then a deviation above the average abundance for that taxa is represented with a blue bar, and a deviation below is represented with a red bar. So you can see during the 2016 La Nina year that nearly all of the taxa are represented with a blue bar. So that increase in number is not only due to one specific family overwhelming the net tows that year, it really is across the board. And we can also see diversity increasing. This is a rarefaction curve. As tows increase, we can see the number of taxa increasing, and there's a stark contrast between La Nina and then the ENSO neutral and El Nino years, so many more families were found. I also wanted to investigate how fish family assemblages or groupings change with ENSO states, so which types of fish are found together consistently through that year. So we found several groups that overlapped between some years, the most interesting of which are ingralids and scombrids, which are tuna and anchovies. And this is really interesting because not only are both the targets of some very large Pacific fisheries, but they also have an interesting relationship to one another. And it's been suggested that tuna, adult tuna, which feed on adult anchovies, both are very active spawners. So the appearance of the two together could be due to the fact that the larvae are just being produced in the area that the adults were once in, or it could be due to the fact that larval tuna are actually feeding on larval anchovies. So we're looking to investigate that further. And for this graph, I once again wanted to point out how different La Nina is, this center graph, from El Nino or ENSO neutral. So everything above this black line here, these bolded taxa names, are things that are common to all 3 years. And we can see with these dots, which represent the number in abundance at that tow site based on longitude, we can see that it's much more evenly spread across longitude for each of these families. And not only that, but with the families that are represented below this black bar, those are families that are specific to this year or only 2 years, and we can see that the number of those is increasing as well. So overall, this is so incredibly important to continue studying because El Nino events, as they're increasing in intensity and in frequency over the coming years, and we've seen how that results in a decrease in the individual fish larvae and a possibility that a population won't be viable enough for self-replacement in the coming years. We know that as these intense record-setting El Nino events are becoming closer together, that there may be less time for these populations to reestablish and replenish in between with the increased nutrients and cooler temperatures associated with La Nina events. So I also think that this could be very interesting as a metric for how sea surface temperature overall impacts fish populations in the context of global warming sea surface temperatures. And then additionally, when we start to think about the efficacy of marine protected areas, and of course we want to continue pushing for more marine protected areas to at least limit the anthropogenic impacts, but we have to be very mindful of how we can also attempt to mitigate some of these larger scale climate impacts. And a big thank you to not only all of our sponsors, because this is a really large project spanning multiple institutions, but also to my advisor, Dr. Rojan, and everyone in my lab. We had to sort through so many jars of plankton. I could not have done it as a one-woman show. It's so helpful. So yes, thanks to everyone involved. Does anyone have any questions?
So I was just curious specifically about like the fish that you found, like because climate change is going to inevitably like warm the entire Pacific Ocean, and like the La Nina and like the ENSO cycles are going to just become like completely just not cyclic anymore. I was wondering how many of those fish that you were studying were like, if you know off the top of your head, and like if they were under some kind of protection, or if they're endangered or almost at extinct, or at any kind of like danger level.
Yeah, I only went to the family level here, so I think a lot of that like species endangerment level would be more specific to groupings, but I think another important aspect to consider is how many of these are important fish for large-scale fisheries and how that has an impact as well, because you can protect or like close a specific area to fishing, but it's the ocean and the fish are going to swim wherever they want to. So yeah, I think it definitely has to have that consideration as well. So I'm not sure if any specific, because I didn't ID to species level if they were endangered, but they're definitely at a higher risk because of this.
Does seeing the before and after pictures of the protections of the PIPA give you confidence that other areas will be able to be protected with that much, success? Or what do you think about that?
Yeah, I think it definitely does give me confidence about that. It's a pretty amazing graphic to see that and to see that it was actually effective. I know that a lot of protected areas have issues implementing policies that can be monitored with enough funding, but the Phoenix Islands protected area is a well-funded and well-set-up, well-managed location. So I think that the fact that we saw such a drastic change in such a small amount of time, because it was just between 1 year that those images were taken, I think that bodes well as an example to set for other protected areas. Yeah.
So did you see a difference in larvae numbers between ENSO and El Nino years, or normal years and El Nino years, or just between La Nina and the other two?
Yeah, we see a pretty big difference between all of the years here. 3,400 fish in El Nino, 4,300 in La Nina, and then 3,000 in ENSO neutral. So I think the important part here is that this is only 3 years of data, 1 year for each ENSO state. So as I mentioned, this is part of an ongoing study. So I think as we continue to bring in more data, we can see if those are repeatable. For the sake of this project, I think that this difference is definitely large enough to start getting the wheels going about why things are the way they are. It makes sense in terms of nutrient availability, but I'm looking forward to seeing what we get with the next few years as we continue this research. Julia.
This may be due to more about like research logistics, but relative to other like phytoplankton species, how big are these fish larvae that you're looking at?
Yeah, so most of the fish larvae that I deal with are roughly 2 to 3 millimeters in length. There are a lot that are smaller than that, and as they get smaller, they get much harder to ID. But I don't often get anything above 5 millimeters. So it's all done through a microscope. Yeah.
So do you think that the difference in the La Nina and the previous, was it an El Nino year previously?
Yes.
Do you think it has to do less with the fact that it's La Nina and El Nino and more to do with like maybe a rebound effect of like they were depressed in El Nino and wow, it's a really good chance to come back in La Nina, and maybe in future climate cycles, if we don't see the same sort of yearly like pattern, that you wouldn't see that same trend?
So Kai's asking about whether the spike in fish numbers and diversity is in the La Nina year is potentially a rebound from the El Nino year, right? Mm-hmm. So I think that could definitely be part of it, as the La Nina event is pretty much essentially a rebound of the system almost overcorrecting. So I think that could be a large part of it. I think a lot of it, I've been thinking recently, as it was a weak La Nina event, whereas earlier it was a very strong El Nino event, I'd be eager to see the effects of a stronger La Nina event. So I think that that could be part of the answer. I think what it's going to come down to is identifying which specific environmental variables could be responsible for this spike in numbers.